Hey guys, it's time to start on chapter 10. We're going to talk about the biology of the genome. A lot of this is going to be reviewed for you guys from A&P, but I'll still go through it and try to make sure I explain everything in as much detail as you're going to need. As far as genetics itself, that's the study of inheritance and things that you can inherit, traits. And these guys, these traits are actually expressed. We're going to find out later that just because you have genes for something doesn't mean that it's going to be actually used. So, in our study genetics, we're interested in what's actually used primarily, although the others are important too. The genome, that's all your DNA, the entire thing. All the genes and all the nucleotides and in the order that these guys are in. Okay, recall that DNA and RNA are nucleic acids. These guys are chained together using dehydration synthesis. And these long chains of nucleotides are polymers. And the individual units of DNA or RNA are called nucleotides, but they're monomers. DNA is a polynucleotide. That's kind of obvious. Okay, let's talk about what is a nucleotide. Well, you got three parts, three separate molecules joined together to make this one big molecule. You've got the nitrogenous base. You've got a sugar that has 5 carbons in it. And then you got a phosphate group. And the sugar and the phosphate are what actually holds together the individual strands of DNA, which we'll see in just a moment. And now it's a moment. Okay, so here is our nitrogenous base. This is only one of 5 possible nitrogenous base. This particular one is only used in RNA. Okay, if this were DNA, that would be a T instead of a U. But that's the nitrogenous base, and if you look, there's nitrogens. This is a 5-carbon sugar. There's 1, 2, 3, 4, and then one up top. That sugar happens to be ribose because this has an OH. If that were DNA, it would be just an H, but otherwise it's going to be basically identical. Now, we also need to make sure that we understand now, this is carbon number 1, this is number 2, this is number 3, this is number 4, and this is number 5. You need to know those because it turns out that these guys are going to join each other 5 to 3, and we'll talk about that in the appropriate place. And then finally, this is the phosphate group. It's a phosphorus with 4 oxygens. This guy's got a double bond, and this guy's actually got a negative charge. So that's a nitrogenous base, and there are 3 others for RNA, A, G, and C. And then for DNA, it's going to be A, G, C, or T. So here's some of the other nitrogenous base. Adenine, thymine, the one that's in DNA as opposed to uracil, the one that's in RNA. Guanine and cytosine. Now these guys are attracted to each other with hydrogen bonds. Remember, there are partial positive charges on those hydrogens because the atoms that they're sharing electrons with don't share equally. They pull more toward the stronger atom. So in any case, these are between adjacent strands, which I have one strand of DNA and another strand. So that double helix, what holds this together is those hydrogen bonds. If you notice, between A and T or A and U, we have 2 bonds. Between G and C, we have, is that right? 3. We should have 3 there. Oh, there it is. They just didn't put it up there. Okay. But in any case, there's our 3 bonds. And again, there's our carbons numbered. And we're going to get to that in just a second. So there's the rest of the nucleotides. Now, the 5' end is the end with the carbon number 5 facing this way. So on one strand, it's going to be facing this way. On the other strand, you can see down here, it's facing that way. Now, we also said that there's a number 3 carbon. See how that 3 carbon there is joined to the 5' end? That's the carbon number 5 right there, guys. So that phosphate joins to the 3rd carbon there. And so we call this a 5 to 3' end. This is the carbon number 3 right here. So this would be our 3' end. Now, whatever, if it's 5' here, then it will be opposite on the other strand. That's just the way it works. So that's why the 5 and the 3 are so important. And everything that this guy does, he does in this direction, 5 to 3. So you really need to know that. So here they've just taken all this and just replaced it with a bar, and they're showing you the bonds. As a matter of fact, that's the close-up of these guys right here. So these guys are going to be the double-stranded helix, which makes up DNA. Okay, guys. I think that's everything we can glean off this page. Of the DNA, C and T are going to be called pyrimidines, and A and G will be purines. And if you notice, the pyrimidines only have a single only has a single ring, whereas adenine and guanine have double rings. So anyway, pyrimidines and purines, that's stuff you got to know. Okay, here again we're showing you that in DNA the sugar has an H there, in RNA it's an OH. There is no other difference between these two guys. So there's our thymine and there's our uracil again. And if you notice, these guys look a lot alike. As a matter of fact, this is their main thing. There's an H here, but there's a carbon with 3 H's there. That's the difference. Thymine, T is in DNA, U is in RNA. Now, there is another difference. There's 3 differences between DNA and RNA. DNA is double-stranded and RNA is a single strand. That's one difference. DNA uses deoxyribose and RNA uses ribose. DNA uses thymine and RNA uses uracil. Okay, so what is RNA? Well, it turns out this DNA hangs out in the nucleus or the nucleoid region in a prokaryote. And this guy is not allowed to leave. And so if we need to use him as a recipe book, a cookbook, to make a protein, then what we have to do is we have to make a copy of that DNA, and then that's what goes and gets with the ribosomes and makes the protein. Again, all of this should be reviewed from A&P 1 for you guys, and I know all of you have got to or have had to pass A&P 1 in order to be in this class. In any case, so that's our main differences, and this guy is the RNA, and specifically I'm talking about mRNA, messenger RNA. He's going to be the one that we use to actually make the protein. He's going to be our working copy. So this guy is like a reference book, and this is a working copy. Aside from the messenger RNA that we talked about, which is a copy of the DNA, We also have a ribosomal RNA. It's RNA along with the protein that actually makes up the ribosome. And then we have, that's our RNA, transfer or tRNA. This is the guy that's actually going to bring the amino acids to the ribosome in the specific order that the mRNA calls for it. Okay, let me just say this. When I make an RNA copy of a DNA, that is called transcription. Okay, when I copy my DNA in order to do mitosis, that is replication. These terms have to be kept straight. I'm saying all that because now I'm going to take that messenger RNA and I'm going to put it with a ribosome and make an amino acid or make a polypeptide. Could be a protein, depends on how many amino acids we put on there. And this is called translation. Okay, this has to be kept straight. Now, this guy starts at the 5' end. That's the way we start on everything. It's 5'. But that's the way we do replication. That's the way we do transcription. It's the way we do translation. There are three sorry, I'm trying to think of this. There are three separate parts. They are the E, the P, and the A. The A is the arrival. So here comes tRNA. He's carrying a amino acid. Now this guy, I'm just going to make up some letters. Let's just say this guy is ACA. This guy will be UGU because we know that C goes with C goes with G, and A will go with T in DNA or U in RNA. That's how we know that we're getting the right amino acid. These amino acids have got to be strung together in the right order. If they are not, if a wrong amino acid gets there or gets skipped, the protein could be dysfunctional and not work at all. That's what happens. If you guys recall, sickle cell anemia is only one amino acid different than normal hemoglobin. Okay. See what we've got here. Ribosomes. I don't really think there's much here that I need to actually go over. You guys can read this for yourself. mRNA is very short-lived because after it's been translated, it's going to be cut up so that it doesn't cause us to make any excess protein, because it'd be a waste of energy and of time and of resources. And the body's pretty good about not doing that kind of stuff. I don't think any of this on here. I've already either told you about it. I told you RNA is part of the protein or part of the ribosome, 60% by mass, and the location where the mRNA actually binds to it. All right, let's continue on. Okay, structure, the mRNA, short, unstable, single-stranded. It's a copy of the gene from the DNA, and it's what the ribosomes use to make sure that they put the amino acids in the right order. This guy is a lot longer lasting because he's actually part of the ribosome, and so he needs to be persistent. This guy is going to make sure that the mRNA lines up correctly and that the tRNA is going to line up with the mRNA in the right order and the ribosome, and he's actually going to be the one doing that dehydration synthesis that is going to result in a peptide bond between these two amino acids. tRNA, this guy looks kind of like that. And here they've got a set of nucleotides called an anticodon. That's the one that's going to match up with that section of the mRNA. So if this were AUG on the mRNA, then that would be UAC up here. And the guy with the UAC carries methionine. In bacteria, that's F-methionine, but basically the same amino acid. So the section of mRNA that's labeled AUG, it's also our start codon, but it specifies a certain specific amino acid, and no other amino acid will do. Okay. Everything, all our proteins, the recipes are contained in the DNA. And it's the sequence. It's those ATCs and Gs in the DNA, their sequence, the order in which they are. I may have 2 A's, then a T, then 3 G's, or it doesn't matter. That is the genetic information. And those segments that code for a certain protein or polypeptide are called genes. Proteins, enzymes, and a lot of those are proteins. All the genes in the cell, we've already talked about genome. We also call it a genotype. Now, the genotype versus the phenotype. Genotype is what your genes say you are. Phenotype is what you actually are. So looking here, you see this bacteria, Serratia marcescens. Now at 28 degrees C, this guy will have red pigments on his plate, but in normal body temperature, those things are not present. And so he's got the same genes to make that red pigment, but it's only expressed under certain conditions. So, and even in humans, we don't have all our genes turned on at the same time. We only turn the ones on that we need. If you're at home, you don't necessarily have every light in your house on at the same time. You only turn on the ones that you need for that period of time. The rest of them are turned off. And it's kind of the same way with genes. So whatever genes are being expressed at any point in time, that's the phenotype. That's what a phenotype is. Now, there are some genes that are always turned on. For example, in my house, I leave a light on all night long. It's usually just a lamp in the living room, but we leave it on all night long. For genes, these will be constitutive genes, housekeeping, because the things that they code for are things that we need all the time. Some things we don't need all the time, but some things we do. Gregor Mendel, he's a monk in Austria that began, or I should say, furthered the study of genetics. He was crossbreeding different types of peas in his garden, and he was keeping careful records and was able to find that they followed a pattern. And apparently he had a background in math and physics, chemistry. And he hypothesized that there are things that can be inherited from our parents, and what he called heritable factors we're calling genes today, and that these genes are passed on generation after generation. And something like what he was doing was the color of the flowers, some red, some white. And he called that a character, and of course we would call that today a trait. So, well, yeah, I think he had red flowers, but we'll go with this. So you have a purple flower and a white flower, and you take these two plants and you cross-pollinate them. In the first generation, all the plants that grow from this cross-pollination will have purple flowers. And then these 4 or however many purple flowers, they will cross-pollinate each other without our help. When they do that, 3/4 of those purple flower, 3/4 of the flowers will be purple and 1/4 will be white. This has to do with the concept of some genes are dominant. So let's say this guy's got 2 copies of a gene, right? There's the purple flower and here's the white flower. Now the white flowers are recessive. In other words, if there's a P anywhere, he's going to be the one who's going to call the shots. So you cross these guys, so the first generation are all going to have one purple gene, which is dominant, and one white gene, which is recessive. Now, you take these guys and crossbreed them. That's what we said we did there. Look what you're going to have. You may have one that has 2 purples. He's definitely going to be purple. You may have one here that is purple and white. He's going to be purple as well. You may have one here that is purple and white. He's going to be purple. But looky there. You have 2 recessive genes, and that's going to give you a white flower. That's all that we're talking about here. Okay, how's our chromosomes organized? Prokaryotes are usually circular. Ours are linear, and they're really kind of spread out like a spool of thread off, or a thread off a spool. Most of eukaryotes, we've got 2 copies of each chromosome, and we call that diploid. DNA supercoiling is when we get ready to actually move this DNA during mitosis. We will wrap it and wrap it and wrap it, and that's what our supercoiling is. It's twisted and all that. When we're not doing mitosis, this guy's called chromatin, and he's just spread out all over the place. And that makes sense, because what if I need this gene right here to make a protein? Well, if he's all tied up and he's in the middle of right there, I may not be able to get to him. So during normal operations in the cell, we're going to have chromatin. Topoisomerases are going to help these guys get supercoiled. And that's what you probably think of when you hear the word chromosome. You probably think of that chromosome there that's getting ready to be split in mitosis. DNA packaging, the DNA binding proteins, these guys are called histones. Even when these guys are spread out, every so often there are histones that the DNA wraps around. Chromatin we've already talked about. So these guys are circular, and they usually only have one, although they can have another one. We'll talk about that a little bit later. This is where they get a lot of their antibiotic resistance and a lot of virulence factors. They have a very small special chromosome, but for the main part, this is their chromosome. One copy, so prokaryotes are haploid. These guys got to do some supercoiling to get in there too. All right, let's continue on. Now, this applies to eukaryotes. So there's a gene. I'm sorry. That's a gene too. In between these genes, we've got DNA that's really just a spacer. It doesn't actually code for any proteins. It's just there to keep these guys apart. Now, that doesn't mean genes can't be back to back to back. They can. But we do have some like this. So when in a eukaryote, when you make the mRNA, you're going to copy that whole thing, or may copy from here to here. So when you do that, you're going to have to actually do some work on that mRNA and cut out the non-coding parts. And those non-coding, well, we'll call those exon. They're excised, and they're going to be cut out before it leaves the nucleus to go and be coded. Extrachromal DNA, we talked about that. Those are called plasmids. That's that little circular DNA that has a lot of interesting features like virulence factors, such as toxins, the ability to make a capsule, or it gives them resistance against certain antibiotics. Bacteria share this with each other, and they'll even share it across species. DNA responsible for heredity. Griffith transformation experiment. Okay. Avery McLeod and McCarty experiment. Okay. This guy, they're using what it was is pneumonia bacteria, Staphylococcus pneumoniae. Okay, so if these bacteria cannot make a capsule around them, then they are not going to do anything. So they injected these mice with these guys that didn't have a capsule, and the mouse was okay. Then they injected the mouse with bacteria that had been killed by heat that had a capsule. Well, these guys are dead, so nothing happens. The mouse was okay. Now, this is the interesting part. They got the uncapsulated bacteria, and then they got heat-killed capsulated, and they put them together, injected them into the mouse, and the mouse died. And of course, they took some of the bacteria from the dead mouse there and injected it into this mouse, and then he died as well. So what happened? Well, those dead bacteria eventually broke down, and some of their DNA floated by. These guys got that DNA and learned to make a capsule. That's what happened. So we know it was living bacteria here, and these guys were dead, so these guys had to transform into this in order for this to happen. So there was the control, 3 experimentals, and it proved without a doubt that these guys were capable of transforming. Let's see. Well, you guys can look at that one. I've already read one to you. You don't need any bedtime stories from me. Read through this, and if you and try to make sure that you understand this. If you don't, bring it up at our next session, please, or shoot me an email. All right, guys, that's it for chapter 10. I'll see you in chapter 11.